And good evening. It's a pleasure to be here with you this evening for tonight's Fund a Fellow. The title, as you saw, is Looking Forward, Research and Clinical Care at Georgetown. I want to thank you again for everybody for being with us today for the Fund a Fellow. It's great, gives me a great honor every time to be here, and especially thank the development team at Georgetown University Medical Center. Mark and Claire do a phenomenal job of being able to keep us together during these times where we have had to be socially distanced, but hopefully soon things will start to change. And I also want to give my sincere thanks to the founders of the Fund a Fellow Foundation, Janice Buchanan and Rick Shenna, really, because without their vision, without all of this, we wouldn't be here. Just all the fellows that they have helped fund and trained over the years is amazing, and I just really thank them from the bottom of my heart for their vision, for putting together this Fund a Fellow Foundation to make sure that we are training a generation of very able-bodied neurologists that know how to take care of patients with different movement disorders, especially the Parkinson's and the Parkinson's-related disorders, and now with the dementias. So we're upon a new academic, ending another academic year. So a new academic year will begin in July. So a new beginning in July. So for me, it's a kind of a time of reflection a little bit. What have we accomplished this past year? What has happened this past year? And yet, it's been a challenging year, but at the same time, we've accomplished so much in different settings and in different ways. We have several different fellows finishing. So we're sad to see Dr. Nakano, Dr. Stephen Nakano, and Dr. Amelia Carwin, Dr. Zara Farzel, who are going to be leaving, but they're going to take with them an enthusiasm and a fund of knowledge that's going to impact thousands of movement disorder patients and thousands of Parkinson's patients all across the country, in California, in Colorado, in Detroit, Michigan, all these areas where there's a huge need to have movement disorder specialists, and that's what we have accomplished over this past year. We have trained these new fellows. So it's late spring, early summer. It's almost like a new beginning. It feels out there, as you can see. The vaccine seems to be making a difference. We see the coronavirus pandemic. The number of new cases continues to decrease from week to week. Since we started vaccinating, we're almost there at that 60 to 70% of the population here in the US. We're seeing those numbers finally come down. So hopefully we'll see a little bit of that herd immunity that we've been talking about. And we can see even tonight, if the weather gets a little bit better, the Nats will be having their first game where there could be a full capacity. And if you've been vaccinated, you don't have to wear a mask. So a lot of good things to come. I think hopefully in the future, we're going to learn. And if things do continue to do well, maybe our next Fund a Fellow, we can hug again, or we can see each other again in the same place and share a meal together. So again, thank you all for being with us today. And I'm very happy to introduce my panel to talk about looking forward. But I want to do a little bit of reflection before we start to look forward where we're headed. But the first person that I'm going to have is the Associate Professor of Neurology. He is my esteemed colleague, probably one of the individuals that has just taught me so much over the years, and I've been so lucky to work with him since 2013. Is Dr. Charbel Moussa, who's really our head of the Translational Neurotherapeutics program. He is the lead investigator at the Parkinson and Dementia Lab, and it's just probably one of the most influential people in my life, and has taught me so much. And it's just great honor every time he joins me here in the Fund a Fellow platform, because he continues to teach not only me, but my whole entire um, group. He is a leader, and his fund of knowledge and his education, not only for the fellows, but I think for all of us who work with him in the Department of Neurology, are lucky to work with him on a daily basis. So welcome, Dr. Moussa. Always happy to see you here. And then our other panelist.
It's overwhelming. Thank you, Doctor.
And our other panelist is he's like another brother to me. So I always say Dr. Moussa is my brother from another mother, but I have a younger brother, Dr. Torres-Yaghi, who's the director of the Parkinsonism and Dementia Clinic. And he continues to be extremely influential in developing that clinic. And we're going to talk about, because I think especially when it comes to the dementias, what an exciting time. And we'll get the viewpoint of both of these professors of what the new Biogen drug means, and what does that mean to the neurodegenerative disorder. So these are some of the topics that we're going to be talking about. But first, let me first start off by asking Dr. Moussa a question. And just, can you reflect a little bit? First, tell us a little bit about what is the Translational Neurotherapeutics program, and what do you see as some of the highlights that we've been able to accomplish this past year?
Yeah, the Translational Neurotherapeutics program, is unique in its mission. It's basically discovering a drug and discovering therapies in the lab for neurodegenerative diseases, bringing them to the clinic, study them in the clinic. And this is really my passion, and taking them from the clinic back to the lab, where we can actually benefit from both the preclinical and the clinical research experience. We've in the past year, we've introduced five new potential drugs that we think, especially one of them is we just published we just submitted the manuscript last week. This is a drug that we especially think is important for Parkinson's disease. Some of the drugs are also important for other than Parkinson's disease, but it's important for Parkinson's because it would be the first class of drugs that would really have a dual effect in both cancer and neurodegeneration. There hasn't been a drug developed like this before. It's a drug that enters the brain, and when it enters the brain, it does two things. It helps clear proteins from the brain, proteins that are misfolded. And on the other hand, we think that the drug can reduce the level of inflammation that we see in the brain. So we've done a lot of work despite the pandemic. At Georgetown, a lot of work has been outsourced, where we thought that would increase the tempo of our research and increase the efficiency. And also clinically, we have just completed enrollment for the bosutinib clinical trial in lower body dementia. We are enrolling right now for the nilotinib in lower body dementia, and another drug called K0706, also in lower body dementia. And we enrolled our last patients for Huntington's disease in the nilotinib study. So we hope that hopefully next August, we're going to have kind of top line results from our bosutinib study in low body dementia, and we're going to move on to further trials. We've also published a paper. It's now accepted in Neurology Genetics. It's a paper, the first of its kind from the previous phase 2 study that we did in Parkinson's disease. This paper is really the first paper that shows how longitudinally over the course of the disease, blood vessels change in the brain, if there is any vascular changes or vascular damage. Previously, this topic was not really paid attention for, by the movement disorders field, because depression is like, yeah, you lose dopaminergic neurons, you lose dopamine producing neurons, you develop like motor symptoms. There has been more attention paid to this hypothesis in Alzheimer's disease because of the effects of hypertension and high cholesterol and other cardiovascular problems that would lead to, if you like, reduction of cognitive abilities and functional abilities in patients. We've shown in this paper that in Parkinson's disease, in patients who have contributed to our phase 2 study, we've done some state-of-the-art, analysis called next generation whole genome sequencing of the cerebrospinal fluid. We looked at regulators of gene expression called microRNAs, and this microRNA showed two things. One, they confirmed our preclinical studies and also our clinical data showing that there is an increase in autophagy, or the brain cleaning, if you like, machinery. But also over the course of the year, the patients who were in the placebo, there was a clear evidence from what we call genetics, or it's a field called epigenomics. There is a clear indication there that over 1 year, there was increasingly more deterioration of the blood vessels, particularly small blood vessels, or what we call the blood-brain barrier. However, in patients who received nilotinib and who did better over 27 months, there was a high correlation between a stabilization of the symptoms and prevention of, if you like, damage or impairment of the brain blood vessels. So that's in general what we did last year. I might have forgotten something, but that's a lot.
It is a lot, and the one thing I probably have seen Dr. Moussa probably the busiest ever in the last I think year and a half, especially during the pandemic. He's probably been busier than I've ever seen him before. It's been amazing, the productivity, and we were luckily able to keep a lot of these trials open. We were shut down for a little bit of time, but we were able to finish off the 2-year extension study of the Parkinson nilotinib Parkinson study. And as you said, we not only saw the long-term benefits in patients who are nilotinib 300 milligrams a day, but also those genetic influences in stabilization of blood-brain barrier and even glucose metabolism, et cetera. So we continue to learn a lot about these disorders, and we're looking forward to the next phase, understanding how the drug bosutinib works in Lewy body disease, and we should have those results in August, and we just finished completely enrolling in that trial. So Dr. Torres-Yaghi, you heard Dr. Moussa talk about how drugs like nilotinib or bosutinib, the main way that they work is by enhancing autophagy. How he normally describes this, it clears these proteins by enhancing lysosome proteasomal degradation. So they get rid of these proteins. So it's like turning on a garbage disposal is his phrase that he always taught us. So he turns on and off that garbage disposal every day by giving these medications, pulsatile every day. And now the cell is able to clear these proteins and protect itself and get rid of inflammation, et cetera, by getting rid of these toxic proteins. How does that differ from the new drug that Biogen just got FDA approval for? So how does that drug, what's the difference between the Biogen drug that was just recently approved by the FDA and the drugs that we've been working with through the Translational Neurotherapeutics program? And what are some of the concerns? Because we see some mixed reviews out there when we hear reports from CNN, et cetera. So can you tell us a little bit about it? How are you going to talk to your patients in your clinic, especially somebody who does see a lot of dementia?
Thanks for asking that question. It's so pertinent to the times right now. There's a lot of excitement behind one of the up until now, one of the hardest diseases of our lifetime and lifetimes before us to be able to conquer. And the idea as of Monday that we may have a therapy that can potentially modify the condition or slow decline and improve symptoms, not only hopefully will open the floodgates to research like the ones Dr. Moussa and Dr. Pagan have been talking about, but also a host of different therapies. When I was a fellow and I was soaking it all in and following Dr. Pagan and Dr. Moussa all over the place, they were nice enough to teach me. I would learn from them just walking down the streets of different conferences in different cities. We would enjoy our time together, but at the same time, I was learning. And I still remember thinking to myself, say, how can a therapy like the ones Dr. Pagan and Moussa were talking about, cancer therapies work in Parkinson's disease? And they would both say, it's like the garbage disposal of the cells of the neurons being turned on so that they can eat up and degrade the protein that's accumulating and causing the dysfunction. Now, the key to answer your question, Dr. Pagan, is the garbage disposal is inside the neurons, right? And so what these medications through our Translational Neurotherapeutics program, what they do mechanistically is remove the toxic proteins, like you mentioned, within the neurons. That's called intraneuronal protein degradation. So within the neurons, if we're able to use oral therapies like the ones we mentioned, the medicines are taken once a day, they're oral, they're in pill form, so they're swallowed, then the goal is overall to be able to reduce the protein that our patients that are so brave and participate in the clinical trials, when we look at their cerebrospinal fluid and we measure the different biomarkers, levels of dopamine, levels of alpha-synuclein, the protein, we're actually monitoring those protein levels within the cerebrospinal with lumbar punctures. We're actually able to see that there may be a clinical correlate, an improvement of clinical symptoms, and a reduction of that protein. And so the mechanism, we think, is based on intraneuronal removal of proteins. That differs from the Biogen medication called aducanumab. The aducanumab, actually, the way it's administered is through an infusion, so an IV therapy infusion on a monthly basis. And the thought is that it's actually targeting the protein extraneuronally, so outside of the neuron. There's still protein that develops throughout the brain, but not necessarily it's not necessarily targeted by the infusion that's given monthly, the inside the neuron protein that accumulates. The other issue, and the reason why we are optimistic, but at the same time cautiously optimistic, as Dr. Pagan always teaches us to be optimistic, but always use science and reasoning to kind of think through things, make sure that everything's validated. The major concern was that edema or swelling of the brain as a result of the actual mechanism of action of the infusion trying to degrade and attack the protein that accumulates in the brain then may lead to inflammation. And so constant monitoring is necessary. So it is somewhat of a breakthrough in many ways in terms of potentially its effect that the Biogen medication may have on the body and on the brain. But we have to be very cautiously optimistic and make sure that it's a safe medication, given the fact that based on the clinical trials, edema is something that could happen.
So we're going to be cautiously optimistic and see how this particular drug, but this drug where it's outside of the neurons, where the trials that we've been working for is inside. And Dr. Moussa, why does brain edema occur? Why with this drug that they're basically antibodies against beta amyloid? Why does edema occur?
Yeah, that's a very good question. So there's like amyloid is the protein is very sticky, gets out of neurons, it accumulates in between neurons in the brain, and it forms the sticky materials we call plaques, or the plaques in Alzheimer's disease. So the reason why this protein accumulates in plaques is because it is kind of not processed in the right way in the neurons, so a lot of it gets out, but whatever ends up in the plaques is probably protective. What really stays in the neuron is probably the notorious part of the protein, or the toxic part of the protein, and we've known that for a very long time. Now, this protein, or amyloid, or it's a, if you like, precursor protein called amyloid precursor protein, or APP, also exists predominantly in blood vessels. So it's everywhere in the body, including the brain. It forms the wall of blood vessels. So when you direct an antibody towards this extracellular plaques, that antibody has to go into the blood vessels, and from the blood vessels, it has to cross what we call the blood-brain barrier, and it has to get inside the interstitium, or the space in between brain cells, to ligate, or to, if you like, catch this amyloid. And when it catches the amyloid, it actually comes back into the circulation, and it's basically dumped out of the body via excretion or urination. Now, once you put that antibody in the brain, you expose not only the plaques that are really accumulated in the brain and causing a lot of inflammation, but also you take down a lot of the amyloids which maintain the integrity of the blood vessels. In this case, the blood vessels become leaky, and leakage of the blood vessels leads to more water getting out of the system and going into the brain, and the brain starts to swell. So that's why with antibody therapy in general, particularly with antibody therapy that we direct towards amyloid, that's why we get edema in the brain, or we get swelling of the brain. This is what we call, or what we know as the sink hypothesis, meaning that the antibody goes to the brain, gets the amyloid. Unfortunately, in about 40% of the cases, we have very strong edema that will result from also ligating and destroying amyloid in the blood vessels, and it gets it gets rid of it via urination.
So a big differentiator between nilotinib and this new infusion is that, did we see inflammation with nilotinib?
We did not, and we're not supposed to see any inflammation with nilotinib, because nilotinib does not really care about the extracellular plaques. Nilotinib goes inside the manufacturing site of amyloid, inside the cell where this notorious toxic amyloid exists, and it destroys it before it gets secreted to build up into plaques outside of the brain. So when nilotinib gets into the brain, it gets two different types of cells. It gets into neurons, where the autophagy is activated, or the cleaning process starts, and it prevents amyloid from being secreted. And the secreted amyloid is also destroyed, because nilotinib also goes into immune cells, and the major type of immune cells in the brain called microglia. It goes into microglia, and it turns on autophagy as well. So microglia can go gobble up all the extracellular amyloid and destroy it. And so in this situation, in these circumstances, that nilotinib has nothing to do with ligation of amyloid. It does not touch amyloid in the blood vessels. What it does, and that's why we think it's universally good for neurodegenerative diseases, because it doesn't really matter if the protein accumulating is amyloid or tau or alpha-synuclein or Huntington's or any other protein. Any protein that needs to be degraded in the brain, that needs to be cleaned from the brain, we clean it via this garbage collection or garbage disposal machine, which is called autophagy.
Yeah, so I think one of the most exciting things that just kind of stepping back and listening to both Dr. Moussa and Dr. Torres-Yaghi, what has been accomplished so far especially since we started working together back in 2013, is, we learned what was happening in the preclinical world from Dr. Moussa when he showed us the turning on of autophagy and the clearance of these proteins in the preclinical, or what we call the bench. We moved it over to the bedside by doing both phase 1 and phase 2 clinical trials in Parkinson's disease, Alzheimer's. We're currently doing it in Lewy body disease. And once we finished that work, we then used the patient. So it was these studies were no longer done in mice, but we saw results from the cerebrospinal fluids of patients with Parkinson's exposed to this drug for over 2 years. And we went back to the lab and we were able to get results teaching us about how these microRNAs and these genetic changes and stabilization of the blood-brain barrier, et cetera. So we've really seen a full circle of both the clinics and the labs working together here. A lot of universities talk about, we want science going from the bench to the bedside. And they talk about that they're going to do this. We're actually doing it, but not only are we going from the bench to the bedside, we're going back to the bench and learning more and how we can advance these drugs. And as a result, there has also been, we've learned a tremendous amount of how exactly nilotinib works. And in fact, Dr. Moussa has discovered new drugs. Can you tell us about some of the new drugs that you have discovered? And and not only the new drugs that you've discovered, but share with the audience the excitement with these new drugs. Where are they at? When are we going to see them in phase 3 clinical trials? And who's going to be responsible for doing that? Can we share with the audience the excitement that we have here at Georgetown University Medical Center?
I definitely cannot be more excited than you, Dr. Pagan. But yes, absolutely. So going back from the clinic, looking at cumulatively, okay, what let's say nilotinib does or what positinib does, we've learned a great deal of information from, in 3 different areas, if you like. First, we've learned about the safety of the drug. We think that we are dealing with very, very safe drugs. So very little attention has to be paid to safety once we move these drugs back into the lab. That would have to be studied, or if you like, more of patient surveillance has to take place in a phase 3 clinical trial. And we're doing that, and I'm going to go into that. The second very important area of learning there has been, we've learned a lot about the preclinical effects of the drug. We've learned a lot about the drug basically inducing autophagy, increasing the level of dopamine in the brain, making the mice learn better, making the mice walk better. But in the lab, we learned actually, as I just mentioned before, we've learned that some regulators, some gene regulators in the brain of humans can actually respond to nilotinib treatment. And not only we've learned the response, but we've learned that actually there is a defect that was previously unknown related to impairment of the vascular system in the brain that now we can fix. And the third thing that we've learned from what we know, what we did, we know called pharmacokinetics, like how much the drug gets into the brain. And once the drug gets into the brain, what fragment of the drug gets into the brain. So the patient takes that drug, in the human patients, the degradation of the drug can be different from the mouse. So we've learned how exactly the drug is broken down, is chopped up. We've taken that specific fragment that got into the brain, and then we started from there. So in the case of nilotinib, we've taken what we call the DORA molecule, or once the drug was ingested, it went to the liver broke it down. One molecule went, or one fragment of nilotinib went to the brain. We've isolated that fragment, and we've learned this is probably the neuroactive fragment. And this is definitely the fragment of the drug that gets into the brain. When nilotinib gets into the brain at 1%, so 90% of nilotinib stays in the periphery, in the plasma, or in other organs in our body. Only 1% of nilotinib gets into the brain. But that 1% of nilotinib that got into the brain is that small fragment. So we took that fragment, we took it to medicinal chemistry on campus, and we did some modification. And we ended up with 12 new compounds, 12 new compounds that are now not, limited in their brain entry. When you give them to the mouse, there is 50-50 distribution of the drug between the plasma and the brain. So we learned how to increase the entry of the brain, of the drug into the brain. The second thing we've learned is how we can develop a drug that now can bypass all the, if you like, safety or unsafety profile of a drug given in the clinic for cancer. And now we can develop a drug that can maintain the blood-brain barrier and lower inflammation. And again, out of a very large library that we developed, 12 of these drugs are now very, very good candidates. And I must say, at least 4 of them are very good candidates to move into different types of neurodegenerative diseases that would involve protein accumulation, that would involve inflammation, and that would also involve impairment of the blood-brain barrier. These drugs, part of these drugs are now, Georgetown University spun out a new startup called KieferX. This is a new pharmaceutical or startup biotech company, which is now tasked with, to study these drugs. Some of these drugs will go to what we call non-clinical development, which means we need to get the safety profile in a number of small and large animals, maybe a rodent and a monkey. Once we do that, we get what we call an IND, or the first investigational new drug. And that investigational new drug will enable us to do the first in human clinical trials through a phase 1 study. Also, with nilotinib, we've actually learned a great deal about the dose of nilotinib. We learned a great deal of where we can take the drug next. And believe it or not, in 3 or 4 years, Biogen accelerated approval of aducanumab is conditional. So in 3 or 5 years, our own phase 3 study, which will be done through Kiefer, will actually produce data of an oral drug that is going to be juxtaposed with that infusion, okay, that, Biogen will be given to patients. The drug may not be, marketed or sold or or distributed widely to clinics, because that conditional approval also involves another phase 3 study to demonstrate the efficacy of the drug. And by the time that trial is finished, then our phase 3 trial, which would involve almost 1,300 Alzheimer's patients, will also finish. And then patients and caregivers and doctors will have to make a decision whether you want to put your patient on an infusion that would, if it proved to be efficacious, an infusion, and an Alzheimer's patient would have to come to the clinic once or twice a month for that infusion. Or if you want to give an oral drug, again, if it proves to be equally efficient, we think it's more efficient, and that's what our phase 2 study showed. It's probably and patients and caregivers will have to make this decision. So we've come a very long way learning both ways. It's a two-way street for us. This is something that other institutions do not do, and we've done it. So we kind of intertwined the clinical with the non-clinical or preclinical drug development, and we are now trying to develop these drugs through a kind of a university-owned partly owned pharmaceutical company.
Yep, it's a really exciting time. So full disclosure, both Dr. Moussa and myself are co-founders of Kiefer, and now it's really up to Kiefer to be able to hopefully deliver and do these clinical trials and move forward in the development of nilotinib, bosutinib, and the dialatin. So it's a real exciting time, and especially comes with the recent approval of this Biogen product. But we're also very eager to see nilotinib and these other products continue to move forward. And moving them outside of the university was definitely the right way to go. And again, working together with the university to form this company has been a real exciting time. So a lot really going on, and the fellows have been part of this the whole entire time. They've been so integral in helping with the phase 1, phase 2 clinical trials, and still with the current clinical trials going forward. But at the same time, there's also excitement in the clinics. So Dr. Torres-Yaghi, can you tell us about some of the excitement to come with the clinics? What are some of the changes you made in the clinics, and what are some of the changes that are to come in the near future? You want to give some good news too about where we're headed to in the clinical world? I mean, because this is the beauty about the Georgetown program. Not only are we working with such great scientists like Dr. Moussa, but and we translate that into the clinics. So what else are we doing in the clinics? What are we developing there?
I think being able to, right now I'm on a Zoom platform, and I'm in the middle of both. Dr. Pagan's over there and Dr. Moussa's over there for me. And I feel like, exactly what they're exuding in terms of this passion for their translational program, for their basic science, bench side research, and then getting right to the bedside. I feel like the bridge really lies in this fellowship, because when you do a Funda Fellow fellowship, what you really do is you're using all of that clinical understanding and that education that you get from Dr. Pagan, but also to think about it with a lens that has very much to do with what Dr. Moussa is teaching you. And so having both of those kind of the inputs into your education really allows for you to become a better clinician. I still remember both Dr. Moussa and Dr. Pagan say, you become a better clinician when you are also a clinician researcher. And you become a better researcher when you're a clinician that understands both. And so they really come hand in hand. And so in our clinic, it's been very, very exciting. We've not only been able to really, really put a lot of our efforts into understanding our patients, understanding their cognitive aspects of their care. We've already talked many times about the Parkinsonism and dementia clinic, but that was just the beginning. We have a cognitive focused examination clinic now. We have all of the fellows are trained in understanding both Parkinson's disease and cognitive changes that might occur, in addition to many of the neurodegenerative diseases. We really do think about all of our disorders that we take care of as protein accumulation disorders, and that's a common theme. We've also started bringing in technology into our clinic. We are using applications that monitor tremor, measure eye movements. We have a different strategy now, especially with the opening of the Center for Neurodegenerative Diseases that I feel so honored to be part of, coming very, very soon. Our goal is to not just be able to treat our patients clinically with medications that are available, but also understand everything that's in the pipeline of clinical research, and become one of the places that is providing a lot of the data to the world about neurodegenerative disorders and disease-modifying therapies like nilotinib and bosutinib and the other compounds that Dr. Moussa mentioned. Furthermore, we've even created, because of the Funda Fellow program and because of other funding sources, because of this importance behind research, we actually started a position within the fellowship for a research fellow position. So someone that is coming in and their entire kind of focus is on research and clinical research, and of course, clinical care, but a research fellow position that we're very excited about.
Yes. So as Dr. Yaghi said, we have a new center that will be opening up in pretty much literally across the street from our current McLean site, a larger location. There'll be a neurodiagnostic center on the first floor, primary care, MedStar Health primary care on the first floor, and we will have the third floor where we'll have the C&D. The C&D stands for Center for Neurogenerative Disorders, and we're redefining brain health. So that's what we're doing with the Center for Neurogenerative Disorders. We're going to redefine brain health with this center. And again, so some of the catalysts for this was also an idea brought by Janice Buchanan and Rick Shenna, and they really inspired us in the Department of Neurology to bring people together that have a lot of similarities. So drugs that we potentially could use in Parkinson's could also be used in Alzheimer's disease, which is the most common neurodegenerative disorders, or Lewy body disease, or ALS, or Huntington's disease, or potentially multiple sclerosis. So we're going to put all of these experts all together in one center with the main focus of improving and improving access and how we deliver clinical care. We got to continue to work on that, improving the clinical care of these patients, improving the education, and continue to educate nurses, doctors, fellows, medical students, residents, and then also have a research center. So it's a full clinical education and research center to redefine brain health. And that's where we're going to be headed. We'll start our first footprint of that in McLean. I especially with this new drug being approved by Biogen, I think we're going to outgrow that space really quickly. But that's where we're headed. That's the future. And what I wanted to do is kind of a reflection this evening, where we go, where have we been, what have we done, and where we're headed in the future. And that's why we're so excited. That's why you see the excitement in our eyes. We see with what we've accomplished with Kiefer, with what we've accomplished with the Translational Neurotherapeutics program, and where we're headed with our new center to redefine brain health. So we've had a few questions come in for question and answer. So one of the first questions that we have, and it seems that there is a Dr. Sackner Bernstein that seems to indicate that dopamine replacement therapy can cause oxidative stress because of the metabolites by producing more dopamine. What do you think about that, Dr. Moussa? And if his hypothesis is correct, does that mean we shouldn't use levodopa? Or we should only be using dopamine agonists? What do you think about that particular question or theory that is being posed?
This is one of the questions that has been addressed or studied. One of the issues that have been studied for almost 50 years in dopamine metabolism. Not to say that this is not a good paper, but I think in the late 80s, some people developed, or some people published a report in the New England Journal of Medicine, and they said dopamine is toxic, levodopa is toxic, we should stop using it. And that was kind of like the Pandora's box into this question. I think 20 years of research have kind of very much settled the idea of why and why not. Dopamine metabolism is not considered toxic, and we know a lot about that. Once dopamine is broken down, it breaks down into two different metabolites. One is called DOPAC, which is predominantly in the intestine or in the periphery. One is called DOPAC, which is predominantly in the brain. And the other one is called homophenolic acid, or HVA, which has very little probably concentration in the brain, like maybe about 15%, and the rest of it is all intestine. So you gotta, and both of these metabolites are not biologically active metabolites. They are not biologically active metabolites, meaning they do not react with any other drug or with any other tissue. So the fact that these are not biologically active metabolites, it means that the active compound called dopamine, which is very active, it can actually, it's very unstable. If you put it in the air for a few seconds, it oxidizes and it breaks down immediately. So the dopamine metabolites is kind of a breakdown, or if you like, detoxification of dopamine once dopamine is metabolized. Now peripherally, we have very little amount of dopamine in the body, you know, like outside of the brain. You know, like we have a lot of dopamine in the gut, we have a lot of dopamine, you know, in the kidneys, but we don't have a lot of dopamine in every other part of the body. It does exist, but predominantly it's in the gut. When you have dopamine broken down in the gut, you really do not have you know, like a biologically active compound. Now, a lot of other people have done research, and particularly like at the NIH, including Bernstein, you know, David Bernstein. He did a lot of research there, and they showed that with some other drugs, if you give dopamine might actually interact with other drugs, and it may cause some cardiovascular effects. He's shown that, but other research, did not confirm that same thing. So there is no clear, there is no evidence that dopamine metabolism causes any toxicity or any oxidative stress. It is actually a detoxification of a very toxic molecule called dopamine if it's there in high level in the body, particularly in the gut, not in the brain. In the brain, we get rid of dopamine very quickly, but in the gut, it can accumulate. It can cause a lot of problems, particularly like gallbladder and constipation and so on. So the fact that it is a detoxification process, that dopamine metabolism is a metabolism, the detoxification process, you know, the side effects of that are minimal. And this has been studied over and over and over for years.
And Dr. Torres-Yaghi, what's a drug that we use that we think counteracts that oxidative stress, so stop the breakdown of dopamine? Because when you, as Dr. Moussa stated, if you break down dopamine, you produce dopac, and a molecule that also comes out of that is hydrogen peroxide. So what drug do we use to be able to prevent that from happening?
You got it. And so just hearing, you know, the way the question was answered, you know, really does help me answer the question you just asked, because if you really understand and if you really learn the science, as Dr. Moussa kind of laid it out, our goal really, when we are thinking about the different therapeutics to offer within the clinic, is to be able to answer a lot of those questions. We might consider in many cases prescribing a medication called rasagiline. Rasagiline is a medicine that actually blocks an enzyme that breaks down your own dopamine. So rasagiline is a medicine that's a monoamine oxidase inhibitor, and it inhibits that enzyme monoamine oxidase, which can break down your dopamine into those metabolites that then break down into potentially oxidative species. And so if you can help reduce the breakdown of dopamine, have more dopamine almost augmented within your neurons due to that medication, what you're doing really is allowing for dopamine to then be sitting around in the neurons for a longer period of time or between neurons. And then it gets taken back up into the neurons, and then it's reused and recycled. And so using therapies that help block enzymes is something that we do quite regularly within neurology. But in this case, thinking about it from a dopamine standpoint is very helpful.
Yep. Yeah. And that's one of the things that we've learned over the years is targeting from different angles. And Parkinson's is much more than just a dopamine deficiency syndrome, but the notion that we should stay away from dopamine replacement, still levodopa is the best medication that we have, and we have ways to mitigate that oxidative stress. We've also learned that drugs like glutathione, which is what is used together with glutathione peroxidase to turn a molecule of hydrogen peroxide to water, is not a viable treatment either, because there's a lot of people giving infusions out there, but it doesn't cross the blood-brain barrier. So a drug like Rasagiin could be definitely much better to use instead. And the ultimate goal is we are replacing the dopamine deficiency by giving drugs like levodopa, but the ultimate goal is to stop the neurodegeneration, and I think that's what we're trying to target, definitely in our Translational Neurotherapeutics program. But I want to thank both panelists, Dr. Moussa and Dr. Torres-Yaghi. As always, you both are amazing. It's always awesome to have you on our panel. I definitely can't wait till we can do this live and together again, and also to meet up with all our Fund a Fellow participants. And again, I want to thank everybody from the bottom of my heart for your devotion to us and for your philanthropy over the years to help able to train the next generation of movement disorder specialists. I'm looking forward to seeing where Dr. Nakano, Dr. Carwin, and Dr. Farzal end up in the future. It's always good to see their journey, seeing them move forward, and looking forward to working together with Dr. Gadani and Dr. Patel, who are going to lead us as being the senior fellows next year and welcome a new class of fellows. So thank you all again for attending tonight, and I'll hand over the baton over to Dr. Torres-Yaghi. Thank you for all your support. God bless you all, and I hope you all have a wonderful summer, and hopefully we'll see each other again in the fall.
Thank you.
Thank you.
I'm just gonna also say, I know we have about one to two more minutes just to close out. I wanted to also thank everybody. This is a special Fund a Fellow because of the time of year. The fellows are leaving, but as Dr. Pagan mentioned at the beginning, they're going all to different places to be able to provide care for other Parkinson's patients and other patients that need more Parkinson's doctors that really know and have understood what it means to be a real clinician, but at the same time understand the research behind everything that goes into the actual therapeutic options that there are available for our Parkinson's patients. Hope is something that all of these therapies that we have at our disposal right now in 2021 is so part of what it is that we try to exude while we're in the clinic. We have more than 25 different medications for Parkinson's disease available right now. And as this, the title of today's Fund a Fellow, when you think about what goes behind it in terms of clinical research, the future is bright. And there is a lot more that this field has to offer in terms of therapeutics. Now, all of what we talked about today was about modifying the disease, halting progression of disease, keeping symptoms at bay. And really that's where we're focusing on. And all of the clinical research that you've heard about from Dr. Pagan and Dr. Moussa, that the fellows are part and parcel for. They are part of that whole process. They're learning it from the inside and out. They're able then to really look at the different clinical options through that lens. I mean, just at the end of this meeting, as Dr. Moussa's mentioned the sink hypothesis, and I reiterated what Dr. Pagan taught me about the garbage disposal. I'll never look at my kitchen sink the same ever again. And so hopefully whenever you think about our presentation today, you think about the garbage disposal and the different medications that we're trying to study to learn more about degrading proteins, removing those toxic proteins, and the sync hypothesis, understanding how it is that drugs are delivered into the brain and the differences of the blood-brain barrier crossing. Hopefully that came through tonight. Thank you so much.